All right, class. So we understand that as long as there is neural stimulation of the muscle fiber, there will be action potential, right, coming down the motor neuron. And as the action potential comes down the motor neuron, there will be acetylcholine released, and that will excite the muscle fiber. Excitation of the muscle fiber will cause calcium release into the sarcoplasm. Will result in calcium release into the sarcoplasm from the calcium release channels. And of course, we will have an abundance of calcium in the sarcoplasm. Now, these calcium will then attach to troponin. There will be cross bridge formation and contraction will be maintained. Now, this cannot continue for long, right? If the muscle is contracted for a long period of time, that will result in tetanus and fatigue. So therefore, the muscle has to relax. Now, we need to understand what needs to be done for a muscle to relax. Well, first, the action potential that was coming down from the motor neuron, that has to go away. And then all the calcium release channels on the sarcoplasmic reticulum, those need to be closed. And all the calcium that is floating around in the sarcoplasm, they need to be resequestered back into the sarcoplasmic reticulum by active transport, meaning calcium will be transported back into the sarcoplasmic reticulum via calcium ATPase pumps, which requires ATP to move calcium against concentration gradient back into the sarcoplasmic reticulum. Therefore, we will not have any more free calcium in the sarcoplasm that can bind to troponin. And therefore, in absence of calcium, the tropomyosin moves back and blocks the active site of the actin proteins, right? So when all these happens, the muscle will relax. All right, moving on to types of muscle fibers. Now, there are primarily two types of muscle fibers, and we will talk about these two. There is a third type as well, but we are not focusing on the third type now. All right. So the two types of muscle fibers are slow red, which is shown in the top panel of the picture, and the second one is fast white, shown in blue in the bottom. Now listen carefully, hear me. If we trigger a slow red muscle fiber to contract, no matter how many times we stimulate it, it would maintain the same force of contraction over time. I repeat, if we trigger a slow red muscle fiber to contract, no matter how many times we stimulate it, it would maintain the same force of contraction over time, as shown by the red lines. In contrast, if we stimulate a fast white muscle fiber, over time the fast white fiber would fatigue and generate less forceful contractions, as shown by the blue lines in the bottom panel. So, slow red fibers maintain same force of contraction. Fast white fibers, they get fatigued over time and generate less forceful contraction. That's a basic difference. Now, why is this? Slow red fibers are called oxidative fibers because they rely on oxidative phosphorylation to generate most of their ATP. In order to do this, they must have many mitochondria and have a rich blood supply and a lot of myoglobin. Now, what is myoglobin? Myoglobin is a protein within muscle fibers that binds and stores oxygen. Additionally, these fibers are small in diameter and have slow cross-bridge cycling period. In contrast, white fibers are called glycolytic fibers. They are larger in diameter because the cells have accumulated a lot of cytoplasmic proteins involved in glycolysis. They also lack robust myoglobin. They lack capillary beds and mitochondria. Because glycolysis does not generate a lot of ATP, this means that over time, this muscle cell will lose their ability to sustain a forceful contraction. So let's summarize the difference between fast glycolytic, white fibers versus slow oxidative or red fibers. Let's talk about this one more time. First, let's address their fiber size. Remember, slow red fibers are smaller in diameter and fast white fibers are larger in diameter. Slow red fibers are called oxidative fibers because they primarily generate ATP through aerobic processes. In other words, they use oxidative phosphorylation. In order to do this, they must have a continuous supply of oxygen. Don't you agree? Their blood supply is very robust and they also have a high amount of myoglobin, as we mentioned before. And the myoglobin is a protein within the muscle cell that can bind and store oxygen. In contrast, the fast white fibers are considered anaerobic. They generate ATP through an anaerobic process called glycolysis. They therefore have less robust blood supply and a low amount of myoglobin. Now remember, in case of the slow red fibers, the rate of recocking of the myosin heads is way slower. And in case of the fast white fibers, the rate of recocking of the myosin heads is way faster. So these are the basic differences between the slow and the fast fibers. All right. Now, where in the body do we find the slow fibers and where do we find the fast white muscle fibers? Okay, your erector spinae. Do you remember the erector spinae muscles? Your posture muscles. These are your posture muscles that help to maintain your posture. Now, the erector spinae muscles are made up of the slow red fibers that can sustain longer contraction over time, keeping you seated on the chair. Where in the body do we find the fast white muscles? Well, while writing exams or taking notes, the muscles of your hands become fatigued, right? Perhaps you put down your pencil and shake your hand a bit. Your intrinsic hand muscles are made up of fast white glycolytic fibers. Are we good? Okay. Now, some general information about different types of muscle fibers. Now, in any muscle, there will be a mixture of slow and fast fibers. Okay. And in order to generate a faster or forceful contractions, the fast fibers will be recruited first. Okay, so the fast fibers help out when particularly forceful contraction is required. And for normal contractions to take place, the slower fibers will be activated first. So for forceful contraction, the fast fibers will be recruited first, but for normal contractions, the motor units containing slow fibers will be recruited. And different people have different proportions of these types of fibers, and there is actually very little evidence that say that training alters these proportions in humans. Okay. Moving on to contractions of skeletal muscles. Okay. First, Okay, before I talk about these, I want to point one thing really, really important. Remember that a mechanical event is always preceded by an electrical event. So you have the electrical event first, which is then followed by the mechanical event. Rupa, can you give me an example? Yeah, why not? You wake up in the morning and you put bread into the toaster, right? In order to have your breakfast. Now, for the toaster to actually toast the bread or the slice of bread, do you need to have electricity? Yes. So you plug in and you flip the switch, right? So there is this electrical event first, which leads to the toaster running and toasting of the bread. So, and this is true for every single thing. This is true for the refrigerator. This is true for your hair styling iron. This is true for everything. There has to be electrical event first before a mechanical action. And the same thing is true in our body as well. There has to be the electrical stimulation for a muscle to contract. Okay? All right. Now, on the right-hand panel, we see graphs, right? Now, the blue lines represent stimulus or action potential or shocks. And the pink lines represent muscle contractions. Okay, or muscle twitches. And I want you to notice one thing here. If you notice carefully on the top panel here, you see that the stimulus occurs first, right? We have the stimulus and then later we have the muscle twitch. Therefore, what do we see here? We see the electrical event first and the mechanical event follows the electrical stimulus, right? Very important concept. Now we will start with the twitch. And a twitch happens when there is a stimulus or a shock, and that stimulus causes a muscle to contract quickly and also relax completely after the electrical shock. So you give an electrical shock, the muscle will quickly contract and then relax completely. That's a twitch. Now what happens as you increase the voltage? You know this. As we increase the voltage, that increases the strength of the twitch up to a maximum value. Now, what if you apply a second stimulus or a second shock immediately after the first, and you don't really let the muscle to relax? What happens then? Well, then you have summation. So in case of summation, what do you see here? We see at the very, very bottom, can you follow the blue lines? We see that there are two stimuli in very close succession. So the stimuli are kind of immediately one after the other. Therefore, you have the first twitch, and the muscle is not relaxing completely. Even before it can relax, there is another stimulation, and then the muscle, and then you have another twitch. And therefore, we see that the second twitch will partially piggyback the first. And this is called summation. And you have also seen this in labs. All right. Now, this is a diagram that you have seen in your lab mini lecture. Now here we see few